Hello everyone, I hope you're well. I'm Nikolas Pontikos. I'm a principal investigator at the UCL Institute of Ophthalmology and a bioinformatician by training. I'm also the co-founder of a bioinformatics startup called Phenopolis. Ismail Mogul, who is a contributor to this presentation, is also my co-founder. Today I'm going to be talking to you about how to develop novel bioinformatics software. So as I was saying, my background's in computer science, and I've worked a bit in industry as a software developer and as in a startup, but I quickly transitioned into bioinformatics, first doing a MSc at Imperial College, followed by 2 years working at the European Bioinformatics Institute in the UniProt team, which is one of the biggest protein databases in the world. Following that, I then did my PhD in bioinformatics at Cambridge University, where I worked on a different sort of biological data set. I was working mostly on flow cytometry data. After completing my PhD, I moved back to the UCL Institute of Ophthalmology, to UCL at the Institute of Ophthalmology specifically, where I've been doing mostly the bioinformatics of rare eye diseases. A lot of my work has been around building databases of genetics data and also web services. And this led to me establishing, spinning out a company in 2018 called Phenopolis. And this is something I'm still doing on the side, but I'm a full-time principal investigator at Moorfields at UCL Institute of Ophthalmology. It's a side project at the moment. And these are just two other projects that I'm kind of working on, which I can talk to you about at our next session. So what is bioinformatics? So different people might have different ways of interpreting or talking about bioinformatics. Another commonly, I think, in pretty good descriptive term for it is computational biology. The general definition of it, which I like, is that it's the application of computer science into statistics, so i.e. software, to the study of biological data. Bioinformatics really is a cross-disciplinary, interdisciplinary science, which includes chemistry, biology, statistics, engineering, mathematics, and biochemistry. It's been around for some time, since the 1950s. Here is the picture of what we think to be the first bioinformatician, Margaret Dayhoff, that started with working on punch card computers up at the time and working on protein sequence, peptide sequence assembly. Bioinformatics really has taken off in the last 30 or so years. In 1990, really picking up with the sequencing of the human genome, which generated increasing amounts of data. And now we're really like in an exponential phase where we are generating huge amounts of data every day. Here's just a word cloud of what people, the words mostly associated with bioinformatics, and you can see that sequencing is one of the most expressed words, gene, protein. So it's really, bioinformatics really is a lot about analyzing DNA and RNA sequencing data. So as I mentioned, if we're looking at the types of biological data, the most prevalent and most important type really in bioinformatics is sequencing data. And sequencing data means DNA sequencing, means RNA sequencing, means protein sequencing. And there's different types of sequencing experiments that you can do to get to look at different sort of, if you have different questions. So you can look, for instance, you can get an idea of the function of the DNA by looking, for instance, at methylation, or you can just look at the variation in the DNA, which is the most sort of traditional use of sequencing. But beside that, there's also all kinds of high throughput biotechnologies, which generate large amounts of data which need to be analyzed efficiently with computers. So for instance, high throughput immunofluorescence, like fish we might have heard, can be done in a high throughput manner. Also, flow cytometry, which I mentioned I did during my PhD, which is looking at immunostaining cells and then looking at millions and millions of cells and basically counting the different types of different cell types. Microarrays, which are kind of the precursor of sequencing, where we have templates, little probes of the DNA, and then based on the binding, this activates a luminescent probe, and then this tells you whether there's presence or absence of a certain sequence. I mean, this can be applied to any sort of stable molecule. So it can also be applied to cDNA, for instance, if you're looking at RNA expression. And also mass spec is also something that, that's been around. I haven't got that much experience with mass spec, but it is also considered bioinformatics. So if we look at sequencing data, what can we do with sequencing data? So there's many applications. I mean, here are just some of the ones that come to mind. Phylogenetics, which is quite relevant this day when with the COVID. We're looking at the lineages of the virus, and look by comparing the sequence of the different virus strains, we're able to see, firstly, we're able to see where the virus kind of came from. That's how we were able to really trace it back and see how it's mutating as well, and how it's being spread across communities. So phylogenetics this is like one very relevant application, but it's been used in the past for inferring basically relationships between different species and looking back at where their most recent common ancestor might have been. DNA variant calling, which is essentially one of the most useful applications of sequence analysis for looking at inherited human diseases. So this is something I do a lot of when looking at rare eye diseases. RNA gene expression, what you're seeing here basically is each of these points is a cell, and this is actually a single cell RNA. So based on the cell, on the expression of genes in the cells that you can infer from counting the number of reads coming from each gene transcript, you're then able to project these cells into a multidimensional space and distinguish different types of cells based on their gene expression. So this is what these sort of clouds of different colors are showing you. And also, I think I mentioned to you just before that with sequencing, you can also look at a function of infer the function, for instance, of DNA by looking at where are proteins binding on the DNA, or what is the sort of state of the DNA. Some examples of sequence analysis software, so bioinformatics software. So if we're looking at peptides or proteins, then we are interested in aligning, looking at sequence alignment, for instance, or we're interested at looking at inferring the function of a sequence based on its similarity to others. So this is what this HMMR is. DNA, there's all kinds of tools to work with short read DNA sequencing, including the SAM tools, which is kind of a suite of tools to go from short read sequencing to assembled DNA to variant calling. And then for variant calling, there's the genome analysis toolkit and a modern sort of deep learning framework called Deep Variant. And then once you've got your variants, your genetic variants, then you want to get all kinds of information from different databases and so on. And for this purpose, you use tools like ANOVA, which is, this process is called, by the way, variant annotation. And so using tools like ANOVA, you are able to go run based on the position in the DNA and the genetic variant. You're able to go to different databases and use that information to get information, to extract data about the genetic variant, such as the frequency in the population, its predicted pathogenicity, uh, obviously, sorry, what gene it's in, which is the most important, what transcript it's in, what effect it has on the protein, for instance. And then for RNA, we're similar sort of process. STAR, for instance, is a program which takes the short read sequencing and then aligns it. Seurat, which is an increasingly popular bioinformatics software, which is used for single cell RNA sequencing. So, I mean, all these tools, one thing is important is that all these tools are open source. And I'm going to talk about this a bit later, but you can find basically the source code for all these tools on GitHub, which is a repository for open source software. So that was sequence analysis. Structural biology is also an important part of bioinformatics. And here are just some applications of structural biology. Protein folding. So from a sequence, predicting how a protein is going to, basically is how it's going to fold and what shape it's going to take. And this is really important because shape is very important for function of the protein. And for instance, if a protein doesn't have the right shape or has a sort of different shape than it's supposed to have, then that can cause a disease. Protein docking, that's really important for understanding how proteins interact with ligands. So for instance, in drug development, because you can imagine a protein and a ligand being like kind of a lock and a key. And so they need to fit together. And RNA folding is another example. So we're talking about RNA, and RNA, based on how it folds, can have an effect on gene expression. So here's some example of structural analysis software for protein folding. You might have seen, I think it was last year, AlphaFold, which is using deep learning AI to try and predict how a protein is going to fold. Then there's software to actually look at the 3D proteins. For protein docking, there is software to look at how the running a protein against a database of ligands. You can see which ligands the protein can interact with. So this is really important, as I said, for drug discovery. And then RNA folding, as I mentioned. You can predict from the sequence of RNA how the RNA is going to loop around and fold. As you can see from here, there's GitHub, but there's also just websites, and this is because a lot of this software is actually runnable as a web service, meaning you go to a website, you type in your sequence, you copy paste your sequence rather, and then you run it, and it will run your analysis for you, and then you'll get the results back. I'm going to talk about this different ways of running bioinformatics software later on. Then we talked about sequence, bioinformatics, structural bioinformatics, system biology is another kind of important part of bioinformatics. And really, here are some examples again of what systems biology can be about. It's usually about networks and relationships between entities. So protein interaction network is an important part of systems biology. Understanding how which proteins interact and which don't. Gene regulatory networks, understanding what the expression of one gene has on the expression of another. Some genes will suppress the expression of the genes, and others will enhance, for instance. And then this really complicated thing here is like molecular pathways. So looking at a sort of molecular level compounds and so on, and seeing how they interact, metabolic pathways, this sort of stuff. Here are just some examples of software. These are actually just resources rather than software, but you can run some bioinformatics tools online through these websites. And then nearly finished with my different bioinformatics domains, but this is the last one, and this is about ontologies, and I thought I'd mention it because it's something that's quite relevant to my research and to my startup, Nopolis, in particular the human phenotype ontology. So this is really about defining relationships between entities, not so much through data analysis, but more having a structured graph on how you represent the world, and you give that to your bioinformatics program can use that to then run its analysis and so on. It's kind of a reference, basically a map of the world according to your computer. So just to make it a bit more concrete, the human phenotype ontology is all about describing human phenotypes. So this example shows you that you can start with a sort of very general phenotype, which is organ abnormality, and then you can drill down to a sort of more specific organ and to a specific type of organ dysfunction. So that concludes sort of the different sort of domains of bioinformatics. There's many other sort of bioinformatics domains, but I'm not going to go into them. Basically, bioinformatics is a huge field. Next important thing, so getting very practical now, is how do you actually run bioinformatics software? So I mentioned all this sort of different bioinformatics software. As I said, some of it is on GitHub, and some of it is is actually a web service. So web services are actually probably the best and most widespread way of presenting bioinformatics software. So it's a website, you go to the website, you upload your data. So as long as your data is not too big, obviously, it works. You upload your data, or you copy paste your data, and then you press a button, and it runs it on the website. Usually it's asynchronous, so you don't like you don't wait there for it to finish. You just you enter your email address, and you will get a notification when the analysis has run, and you'll be able to download your data. I'll talk a bit more about this sort of web services when I talk about Phenopolis in my next session, because it's really what we are working with. Then you can get the standalone software. So kind of the one that people are most familiar with is basically any sort of Windows program or whatever that you download, you get a graphical user interface. So here I'm showing you one called PyMol, which is for looking at 3D protein structures. And you download the Windows program, you install it, and then it's basically a click click interface. That's for most users who don't necessarily know who are not very, let's say, computer savvy. You're limited with what you can do with this because you cannot it's difficult to run it in a sort of batch way. So if you want to run many of these in parallel, you end up having many windows open, and this eats up a lot of computer resources. So it's not very efficient. It's nice to be able to visualize data, but it's not a very efficient way of running software. Then you've got the sort of more, let's say, geeky sort of tools. You've got your command line tools. So not many people use the command line, but thankfully an increasing number of people are getting familiar with it. So command line is, if you've got a Mac, you can open a terminal. If you're running Linux, then you obviously know about the command line already. And on Windows as well, you can get now with Windows, you have a command prompt. But now I'm not very familiar now, but they have a Linux subsystem in Windows, and you get also, the PowerShell is what Windows has. And the good thing about this is it's a much more, as I said, much more efficient way of running your program. You can run many jobs in parallel, And what you can also do very easily is then run it on a computer cluster. So if you want to scale your analysis to large amounts of data, then you can run it on a computer cluster. Now, the computer cluster and the web service are related because a web service is often just a front end to a computer cluster. So this would be, I'll go into that when I talk to you about sort of web services. So where does all this software get written? So a lot of it open source, and that comes from big sort of institutes such as the Broad Institute in the US and the University of California, Santa Cruz. And in the UK, we've got the Sanger Institute and the European Bioinformatics Institute. And then in Germany, there's the European Molecular Biology Laboratory, which is actually one of the first sort of bioinformatics institutes in the world, and the Center for Genomic Regulation in Spain. And at these institutes, you've got teams of individuals working on this software, but also you have a few people that are kind of made a really, wrote the first widely used program. So I was talking about SAMtools, for instance. So SAMtools is basically used for the analysis of all the next generation sequencing data now. And that was one person that started that project, Hengli, who is now at the Broad Institute. Some other software, Plink, for instance, that used a lot in doing statistics on genetics, and that was developed by Sean Purcell. GenomeAD is a huge and very useful database of genetic variants in humans, and ProCheck, which was to check the protein structures, and this was by Janet Fortin at UCL. So there's many other people basically, but these are just some of the, just giving you an example that often it's individuals really that like lead and start the development of something, and it grows into a team of developers working on some software and maintaining it. But it's often the idea of just one person to start with. So if you want to develop bioinformatics software, it's a good idea to have a look at what's already out there, right? So is there any other open source software already that you can use? How easy is that software to run? So if you're not a computer programmer, you might find it difficult if you just have some source code available because you don't know how to compile it and to run it. Thankfully, nowadays, well, firstly, there's binary code available, but there's also what's known as Docker, which I can talk to you more about tomorrow at the next session, which is really allowing you to make it much easier to run your code. You basically download your own sort of what's known as a container, and then you can run your code much easier without having to compile it. Also, the software that's out there, I mean, how good is it? How well does it work? Unfortunately, what happens a lot in research and academia is that people will write the software, publish the paper, but then the software, they won't maintain it at all. It will stop working because the software depends on other software, on libraries, and those will get upgraded, and then it won't be compatible anymore. It won't be extended. So it's a sad reality that a lot of software is basically published and then left to rot, and it's just good enough so that the reviewers of the paper will be able to run it, and a lot of the times the reviewers don't run it, they just look at the results and the output. So yeah, so there's a very all that to say there's a huge spectrum of quality software, of the quality of software. And yeah, based on what's out there already, well, what can you add really, right? And also what is good is to find a niche. So if there's a new biotechnology, for instance, or there's a sort of new analysis method that hasn't been done before, then you can really make an impact by being the first person to develop that software. So the example of Hengli and SAMtools, for instance, Hengli was really at the start of NGS, next generation sequencing, short read data, And he basically developed a whole suite of tools that could work on this sort of data. He defined the data formats and so on. So once you've chosen what you're going to do, then you have to choose how you're going to do it. So some of the decisions you can make are which programming language you need to be using, which databases you're going to be using, how you're going to be storing the data and query the data, and what sort of platform you're going to develop on. Are you going to develop on, you're going to build a web service, as we were discussing, or are you going to build a GUI that's going to be running on Windows or Linux? So programming languages is an important consideration because basically computer science, as you know, is very fast evolving, and programming languages basically get very popular, and then a lot of them can die off a few years later. And this is really what I'm showing you here. This blue line here is Perl, and this red line here is Python. And what you can see here is that this is in the early 90s. Perl started to become very popular, and in particular, BioPerl was very popular as it was the first programming language used for bioinformatics. Here, this blue line here, this is basically the popularity of bioinformatics. By the way, these plots were made using what's known as Ngram, Google Ngram. It's quite a fun little tool. It shows you the frequency of words in the literature. So it's basically parsed millions of books, and then it sees how popular a certain term was over the years. So bioinformatics, as you can see in the early, as I was telling you before, in the early 90s, started becoming very popular with the whole genome, the Human Genome Project, and then started becoming a bit less popular around 2008. And this kind of corresponds a bit to Perl. So Perl gets very popular, and then around, okay, it's a bit later, but around 2008, it really starts dropping, but then Python starts picking up. So this is just to illustrate that, languages come and go. So it's important to choose a language where you are going to get a lot of, that's going to be around for a long time, and it's going to be easy for you to find people that know how to code in that language. If you choose a really esoteric language, it might be great, but there's only 2 people that can code in it in the world, then it's going to be difficult. And basically, which language you choose depends on your expertise as well. But these are kind of languages, BioPython, R, Biointroductor, BioNode. These are languages really that are picking up now for some of them been around for a while, and they're really still going strong for bioinformatics. I mentioned programming languages, databases next. So databases, different types of databases. I don't have as much to say on databases, but really there's the most standard database that's been around since the 70s or whatever is relational databases, which are built around tables. A lot of you probably have used Access, but or heard at least of MySQL, Postgres, and so on. These are really your table-based databases, and then if you want to store your data, you break it up into different tables, and then you can join the tables based on common keys. So this is really, I think, the kind of box standard database. Now people have new sort of databases which are less structured, like object databases, of which MongoDB is an example. These are very good to store objects which have different sort of structures. They're not necessarily basically in a relational database, everything you put in a relational database needs to have the same sort of structure. So once you've defined the structure, it limits what you can put in the database. Object databases allows you to put different things of different shapes and sizes, which sounds great, but then when you want to get the data out, it's still not, you still need to worry about how it's structured. So it's not actually, my opinion is it doesn't save you that much time to have an object database. And then graph databases, which effectively are quite similar to relational databases, except instead of thinking about tables, it's about thinking about graphs. And the nice thing about graph databases is when you have lots of different types, then really it's very easy to add a new type, a new node type, rather than creating a new table in a relational database, but still there's quite a lot of similarities. So once you've got your database technology that you're going to use, I mean, by the way, the same applies for database technologies. Use something that people are using so that you can get support and that is stable. Then you need to think about your sort of platforms. So what are you going to develop for? So as I mentioned, standalone application like Windows or Linux, or are you going to go for web services? So are you going to develop a website? And in which case, then you need to think about a whole lot of technologies such as cloud providers, of which you heard of Amazon Web Services, Azure, and Google Cloud, for example. And also, this will allow you, the good thing about this is all of these providers will allow you to scale. So what that means is if you have like one user on your website, you'll only be using one computer, but if you have like 1 million users, then it will scale and it will create new computers, basically clone the servers, and that will allow you to handle much bigger traffic. The downside is that it's not free, and so you need to be careful about your expenses there. And then if you're developing it for a website, then you can. There's also another consideration now, which is what sort of programming language and web framework to use. And what is meant by a web framework is basically the programming language plus the libraries that come with it. Ruby on Rails is a popular web framework. Python Django is another one. Node is one which is becoming increasingly popular. Right. So I hope that was okay. Like I explained to you, kind of what your considerations when choosing your when building bioinformatics software, what to think about. And now we're going to talk about what if we want to start commercializing bioinformatics software. So this was all good for like open source and so on. But now if you want to start commercializing bioinformatics software, well, firstly, it's a good thing to see what's already out there in terms of companies, right? So big companies, very big companies, BGI, Beijing Genome Informatics, very big company in it, which is sequencing thousands of human genomes a day, massive sequencing power. Genomics England, which you might have heard of, which is basically an initiative which was started by the previous Prime Minister David Cameron to find the genetic cause of rare diseases in the UK. So it's also known as the 100,000 Genome Project. And that's been, sequencing, as the name indicates, over now 100,000 individuals. And so along with the sequencing, they've been developing a lot of bioinformatics software and using existing bioinformatics software as developed by Congenica and Fabric Genomics. A company that you might have heard of a bit more these days is Oxford Nanopore, especially in relation to COVID, because Oxford Nanopore is a company that specializes in doing longer read sequencing. So, next generation sequencing has been short read sequencing, about 200 base pairs, whereas Oxford Nanopore can sequence kilo bases, so thousands of base pairs. And this is important. This is, remember I showed you the phylogeny of COVID earlier on. Well, being able to sequence the whole virus in one go, it makes it much easier than to build these phylogenies. And then, there's also startups such as LifeBit, which have gone through several rounds of seed funding. And there's also very early startups like Phenopolis, which I co-founded, which I will talk to you more about in the next session. So basically, biotech and health tech opportunities. So what kind of software these companies do, now you need to start not only thinking about research, but you need to start thinking about how is it going to be used. and what is it going to be useful for. So really, you need to start thinking about your software as in the long term, depending on what software you develop, it can become a medical device, which means that, it's going to be used to make clinical decisions. And that's going to be, that is, there's a lot, as you can imagine, there's going to be a lot of sort of governance and accreditation around that. So that's something you need to be, to keep in mind. So even if it's like finding a genetic diagnosis of rare disease, that still counts as a medical device. We can talk about it more in our next session at the Q&A. Then you got like medical apps, which you can have on your phone. So like you can imagine, for instance, a app which would allow you to doctors to share pictures, confidentiality of patients, or as you all heard of, like the NHS track and trace app. Then we could start thinking, about digital health systems, so things installed in hospitals, so like patient records, for instance. Applications of AI to various problems, and so on. Also, laboratory equipment, for example. So, like for instance, software to analyze, let's say, images coming from confocal microscopy, or so on. Also, if you're going to start commercializing bioinformatics software, you need to start thinking about intellectual property. And that means that includes software licenses. So people that have your software, like how are they going to be allowed to use it? And also, if your software includes open source software, then probably you will need to pay, depending on the software license, you might not be able to use it for commercial purposes, or you'll have to pay a fee to the creator of the software. Patents and the copyright as well. So patents is really more about methodological patents. So like, for instance, you've developed a method which uses a certain algorithm to do a certain thing. So like using a deep neural network to analyze optical coherence tomography, or to predict the configuration, the structure of a protein, or so on. So that can be made into patents. And then the copywriting, so copywriting of your, you can copyright software, so it's not exact, so no one can copy exactly your code, your source code, and also copywriting of names, for instance, the name of your company. Good organizations, each university has their own sort of organizations to help with this. UCL has the UCL which can give you advice on these things, so UCL Business. And there's also a helpful sort of organization called the UCL Translational Research Office that can also provide you some advice. But UCL Business is the main place, is the most pertinent place to go for getting advice about intellectual property. Software licenses, as I mentioned, you have to be careful. There's quite a few different software licenses. There's software, in the early days of Linux and software development, a lot of stuff was open source, and people would be using open source, and no one would be making a profit from it. But then there's sort of different variants of these open source software licenses with different sort of restrictions. So a good website to check is this one if you want to know what sort of software license to use. And then there's the important, as I mentioned, we were talking about if you're developing medical devices, your software needs to prove that it's doing what it's doing on the tin, and it was built in a certain certified way. So governance is really important, and it's something actually we're going through with our Phenopolis startup at the moment. It's really about showing that you are following the guidelines and that you are using software best practices, that you have audits of everything, and that your software is safe and is built safely. I'm not going to go through these, but this is just, we can talk about this more at the next session, but just some of the governance. ISO 27001 is an important one. This is sort of more of an organization level one, and the NHS data security and protection. If you're working on clinical trials, the FDA ones are quite important for especially the electronic records and electronic signatures ones. Also, ISO 9001, it's all about quality management. It's very important. We can talk about this more in the next session during the Q&A. How do you actually how do you get money to basically commercialize, to pay people, to hire people, and to pay for, for instance, web services as we're discussing. Venture capital is kind of a traditional way of doing it. And you have these biotech accelerators, such as BioStars, government grants. So UKRI, UK Research Innovation, is a good source for that. There's also smaller sort of university grants you can get as well. I mentioned the UCL Translational Research Office. They do small grants as well. I was lucky to get one recently. And also self-funding. Well, actually, this is kind of what we're doing with Phenopolis, because as we're doing it part-time, we still have our university job. We're not jumped completely into full-time startup. Well, we are self-funded at the moment, but you can only go so far with self-funding, really. To get to the next step and to grow and hire more people, you really need to, get a big pot of money. It's very difficult. It's not impossible, but I think it's very, very difficult to grow into a big company from self-funding alone. And some important questions which are very relevant before starting, before even deciding to make a company is, can you work with people you trust? You need to find people that you know well, because it is a marriage, it is a wedding in a way. You're going to be talking to these people every day, and you're going to be trusting them with very private sort of stuff, like with your software, with your money, with your time. So it's important you trust them, and you don't want them to, for instance, take your stuff or your software and then go and work with somebody else, steal your ideas or whatever. There needs to be trust. You need to be a team. You also need to think about what you're selling. Are you selling software? Are you selling data sets? Or are you going to be doing consulting? So that's important, because it's important consideration, because depending on what you sell, you have to consider what sort of people you're going to hire, and what is your sort of long-term strategy. Who will pay for it? And that's probably, I think for me, that's the most important thing, like getting your first customer, or identifying at least your first customer. And also, it's not enough to just have a person that tells you, oh yes, I'm interested in this, I'll pay for it. Find out how easy it is for them to pay. Like in the NHS, for example, if a doctor can say, oh yeah, I'm interested in this, I'll pay for it. But then imagine it has to go through the whole process with the hospital of getting a contract signed and all this, and that can take months. And do you have enough cash or whatever in the bank to keep you going for that long, right? So this is really important, like to find out. It's not enough to just have a promise. You need to do a bit more digging and just find out, just to manage expectations of how long till you can get the money and plan accordingly. Also, identify your competition and what are the barriers to entry. This is a question that people often get asked when they're doing a sort of Dragon's Den sort of pitches. Who are your competitors, and how easy would it be for them to just take your market, basically. And then how would they're thinking about long-term, sort of how will you scale? What is your kind of roadmap, your big sort of your 5-year or 10-year plan? The team, as I said, is going to be very important, and kind of team members that you're going to need. are going to be accountants. You're going to need an accountant because you're going to need to deliver your company finances every year to HMRC, the UK. Legal, because you're going to need to have contracts. You're going to, when you hire people or even contractors, you're going to need contracts. When you sell your software, you're going to need contracts. And then basically, yeah, you'll see the CEO, which can be you, and the CTO, Chief Technology Officer, which can be your friend who's tech savvy. It's usually good to go to, build a company with more than one person, right? Because it's a hard job. So you really need you need a team, really. You need somebody to help you keep on going and to help each other. And then developers, and if you're building software, obviously developers are really important. And I would recommend hiring contractors to start with. But then contractors can be quite hit and miss. We can talk about this more at our next session. And also a project manager, somebody who kind of, follows up on things and makes sure that things get done. That can also be you, but depending on your time commitment, it might be good to have somebody else to do that. And so once you've got your team assembled, then you need to think about the roadmap. So your kind of 5-year, 10-year plan. And it's an important commitment. And how does it fit with your life plans? And what is the outcome at the end that you're hoping for? Are you hoping to sell your company? This is the things that you need to kind of think about, because this will be important on how you structure your company, shares, and so on, and what software you decide to focus on. It's easier to sell a company if you're just developing one type of software rather than many different types of bioinformatics software, for example. And then, yeah, like important considerations, it's difficult. A lot of the time you don't know which way to do. You might try and do both. Stay, for instance, as I'm doing, staying both in academia and also having the job at having a startup part time. But it's difficult, and at some point you're compromising, and some point you might decide to make the jump. Either you're going to stick to academia, or you're going to jump into and full time, and just take the leap and go into industry, into your startup. And that's yeah, an important choice. I think when you're doing that, it's good to give yourself a time and just say, I'm giving myself X years to do this and X amount of money. And if I don't do that, then I will have tried, and I'll move on to the next thing, right? And then, yeah, uh, I think it's good. You should try it regardless of, it's a good thing to try, and everyone should try making their own company at some point in their life. The easy steps is, yeah, find your team, find a good person you can trust and work with. If you're at a university, then go to your institution's business and innovation team, UCLB, and just make sure, everything's done well. I mean, they are, they are there to help you, really. They're not there to tell you not to do anything. They're there to help you. So you can trust them 100%. Don't listen to, some people might tell you, oh yeah, don't bother going to UCLB. They're not, or whatever the equivalent is, they're not going to help. Really, I mean, they're there to, they want you to succeed, right? And they will give you helpful advice as well. Um, and then actually making the company, probably some of you have done that already. It's really easy. You just incorporate your company in Companies House. Just takes a few days, costs £12. Your company will appear on the Companies House website. But once you've done that, then you need to be able to file your accounts every year. And so you do need to have an accountant. And finding an accountant is not too hard. We found ours with through connections, friends. We can talk about this in more detail tomorrow if you're interested. Opening a business bank account, you can do that in a few days if you open a a web-based account like Tide, for instance, is the one we're currently using. Other business accounts can ask you loads of questions like HSBC about how much money the company is going to be making a year and so on, like things you don't really know at the beginning. So yeah, just create it, go with one of those online banks to start with, and then yeah, you can start trading. So we're going to talk more tomorrow, and I'll share with you my experience with about Phenopolis, and yeah, hopefully it will be helpful for you guys. So thanks a lot. Here are just some references which should be helpful. If you're more interested in the kind of history of bioinformatics, have a look at this paper. I mean, a lot of stuff you can find it yourself on Google. Uh, this is the UCL software carpentry. So if you want to learn how to code, this can be a good starting point if you're at UCL. Choosing software licenses, as I mentioned, link to the UCL business and the UCL Translational Research Office. And here's a link to the sort of documentation from the MHRA for medical devices. So just so you can get an idea of like the sort of things to the workflow for a medical device and deciding what kind of medical device your software will be, if it will be a medical device or not. So thank you very much, and I look forward to talking to you at the next session where I'll be answering questions about my experience with Phenopolis. Take care, everyone, and see you soon. Bye bye.